Mic check.
Mic check.
Hello, everyone. Welcome to the 25th Journal Club at Antibuddies. I'm here with Dr. Genevieve Mullins today. Let me tell you a little bit about her. She's a postdoc at UNC Chapel Hill, and her graduate work focused on studying the function of CXCR5 expressing CD8 T cells, which we are going to call follicular CD8 T cells today. Since joining the Milner Lab at the University of North Carolina, she studied the spectrum of CD8 T cell differentiation states in cancer and chronic viral infections. Apart from research, Genevieve also enjoys playing board and video games with her partner. Genevieve, it's we're really glad to have you here today. Before we
Yeah.
start with the science part, I have one question. What's your favorite board game?
Oh, that's hard. I think probably Root. Although I haven't gotten to play it as much lately, because I need a new group of people out here to play board games with.
Oh,
But it's like a little asymmetric game with like little woodland creatures fighting in the forest. It's so cute.
oh, did you say root? I'm going to add that to my list of things to purchase. Thanks a lot. Okay, with that, I'll hand over the mic to you. You can tell us more about your work.
All right, so I'm gonna go ahead and start sharing the screen over here. So today I want to talk to you guys about the work uh, that was published in Journal of Autoimmunity late last year, looking at, as was mentioned, these CD8 follicular T cells and how we identified these cells as localizing throughout the follicle and the germinal center during germinal center reactions, particularly in autoimmune disease, and that these cells maintain both cytolytic and helper functions.
Genevieve, just a second. There is some fan noise in the background. Are you guys able to hear that? Okay, could it be either your laptop?
Maybe. Let me. Forgot that there was the under fan of my laptop.
Okay.
All right. So, first off, I just want to kind of broadly go over autoimmune disease. So, autoimmune disease kind of is a combined kind of disorder. There's lots of different types, much like cancer, and can affect multiple tissues throughout the body. Some kind of more common ones I have diagrammed here. So things like type 1 diabetes that affects beta cells in the pancreas, rheumatoid arthritis that affects the joints, multiple sclerosis affects the nerves. And so these ones are tissue specific, but then there are also these systemic autoimmune diseases like lupus that affect DNA and nuclear proteins, and this can affect tissues and cells throughout the body. And generally, autoimmune disease occurs because there's a lack of balance between effector responses from your B and T cells and regulation from regulatory T cells. So these regulatory T cells are unable to properly suppress the activation and function of these effector cells, and these effector cells, these self-reactive effector cells, then go through and attack and destroy self-tissue. And combined of all of the like 80 some well-characterized autoimmune disorders, these affect about 5% of the world's population. And these are all chronic diseases. And so there's a lot of healthcare costs associated with ongoing care for these. And particularly for the work I'm going to be showing you guys today, I guess my old lab now, focused primarily on using the IL-2 deficient mouse model on the BALB-C background. So IL-2 is the principal T cell growth factor for T cells during clonal expansion following activation, but is also a very important survival factor for FOXP3 regulatory T cells, both in their function and in their development. And what occurs when IL-2 is genetically absent is that you get fewer peripheral Tregs, and those Tregs that remain are decreased in functionality. So there's fewer of them, and they're also less suppressive. Because of the lack of IL-2 for a growth factor for T cells will then switch to using IL-7 as their primary survival and proliferation signal. And then you also, due to decreased suppression, B and T cells will proliferate and activate in increased amounts and produce much more serum immunoglobulins. Particularly for our model, we focus on serum antibodies against red blood cells, as the primary disease that these mice get is autoimmune hemolytic anemia. So antibodies are generated against the red blood cells, and then those red blood cells are targeted for destruction. And so from this model, how we kind of initially started focusing on CD8 T cells in this autoimmune model was doing some T cell depletion assays. So we have our IL-2 knockout mice, and starting at day 8, we were depleting with either CD4 or CD8 antibodies to get rid of either our CD4 or CD8 T cells, and then assessing both mouse survival. And this was going to be a readout of kind of delaying or preventing autoimmune disease progression, as well as readouts about the autoimmune disease. So again, because a lot of this disease is anemia, we look at complete blood counts, particularly to look at hematocrit, and then look at binding of antibodies to red blood cells. And what we found is with our IL-2 knockout mouse model, on the BALB/c background at least, all of the mice die by about 4 weeks old, with most of them not surviving past 3 weeks. When you deplete with CD4 antibodies, you get rid of the CD4 T cells, this survival prolongs out much longer. And so most of the mice are still alive, or all of them are still alive around week 10. And they kind of don't start to succumb until after that. And this we kind of expected and knew, given the role of antibodies in the anemia for this disease. But what was surprising for us was how much survival could be achieved by also removing CD8 T cells. This was not as dramatic as the CD4 T cell depletion, but was still significantly more increased in survival. And so we kind of wanted to keep, okay, what's going on? Why are these, giving better survival? And so, like I said, these red blood cells in these mice get coated in antibodies produced by these self-reactive B cells. And so we basically just then stain these with an antibody against those mouse antibodies in order to assess how many red blood cells are being bound by antibody. And what we found, so generally, we look at IgM and IgG, and these are both really increased in IL-2 knockout mice. When either CD4 or CD8 T cells are depleted, these antibody levels go down. So the amount of these red blood cells bound by antibody decreases, as well as just whole antibody levels in the serum also go down. And this was initially very surprising to us, since although roles for CD4 T cells in antibody production, given their role for encouraging B cell help, was known, it wasn't known for CD8 T cells at the time. And so this was really intriguing to us. And so in order to kind of keep looking at this, those T cells, those CD4 T cells that provide B cell help are generally defined as PD1 positive and CXCR5 positive. So these boxes up here. In our wild type mice, they're totally naive. So generally there's not very many of these cells because they're not activated. But there are a decent percentage of these in our autoimmune mice, which makes sense. They're making a bunch of antibodies, particularly class-switched antibodies. And what we found is that this population also exists for the CD8 T cells in our autoimmune model. Additionally, work that was in our previous paper in GAI, we also assessed other markers of CD4 T follicular helper cells, like ICOS and CD40 ligand and BCL6. And these CD8, what we're going to call T follicular cells, so these PD1 positive, CD5 positive CD8 T cells, also express these other markers of CD4 T follicular helpers, BCL6 being their principal transcription factor. And what we found even in our previous paper was that these cells also retain some markers of kind of more traditional cytolytic cells like perforin and granzyme and CD107A.
Genevieve, I have a small question here.
Yeah.
For the IL-2 knockout mice having more follicular, both helper and cytolytic T cells, is it because IL-2 is normally inhibiting the differentiation into the follicular route?
So that definitely may contribute to some extent. And so we definitely had a lot of reviewers complain about this because they just don't like this model for that reason. And so I think it's either the next slide or something. I'm going to show, we also looked in other autoimmune models that are IL-2 sufficient. And so while the lack of IL-2 likely does aid this to some extent, it is not necessarily the only thing. Let's see. No, it's in a couple slides. I lied. So in general, just kind of the flow through of what I'm going to show you guys today. I'm going to first show that these CXR5 PD1 positive CD8 T cells, are CD8 T follicular cells, arise in multiple settings of autoimmune disease. And then I'm going to show how they localize to the B cell follicle and germinal center in autoimmune disease. We'll also look at the transcriptional profile of these cells, as well as some cytokine production, particularly IL-4 and IL-21, and how these contribute to B cell function. And so yeah, like I mentioned, one issue, of course, with the IL-2 is that IL-2 generally inhibits CD4 T follicular helper generation. And so that might be why we see CD8 T follicular cells in an IL-2 knockout. We also looked in other autoimmune disease models, including another systemic model, which is the fast LPR mice. So these are a lupus model. So they, instead of targeting red blood cells, are going to target DNA and nucleoproteins. We see an increase both in CD4 T follicular helpers and CD8 T follicular cells in this model as well. We don't have it shown here in this paper, but we have also looked in the scurfy mice. So they have a defect in FOXP3 positive T cells. They also have an increase in these CD8 T follicular cells. Where we have not managed to find these CD8 T follicular cells is in tissue-specific autoimmune diseases. So in the NOD and the collagen-induced arthritis models, while these cell mice do have some proportion of CD4 T follicular helpers, we have yet to find any evidence of CD8 T follicular cells in this model. And so generally, this leads us to believe that CD8 T follicular cells arise in systemic autoimmune diseases, potentially due to systemic and chronic antigen stimulation, but do not arise in tissue-specific autoimmune diseases. Okay. That was it for that one. That's more just really to kind of drive home. It's not just IL-2, because people tend to like to point that one out. And so from there, we wanted to really assess, where are these CD8 T cells present, particularly since we've seen that they can influence antibody levels. So antibodies, particularly class-switched antibodies, rely on the germinal center response. And so I have our germinal center here with our light zone and dark zone. The light zone being where the follicular dendritic cells sit. And so when B cells get activated, they can then enter into the germinal center because of expression from CXR5. And then once they're there, they can continue to get antigen signals from follicular dendritic cells and use these signals to enter the dark zone in order to proliferate and undergo somatic hypermutation. And after so many rounds of division, these cells will then return to the light zone in order to receive additional signals. The other key cell here are the CD8 T follicular helper cells. So these are a population of CD4 T cells that when they become activated, express BCL6, which allows for expression of CXCR5. This will allow them to get to the border of the B cell follicle, and once they interact with B cells, this will allow them to fully enter the B cell follicle and the germinal center, where they can then provide signals to germinal center B cells in order to allow this cycle to continue. But can also provide signals to allow these B cells to leave the germinal center reaction as memory B cells or plasma cells, particularly class switched, so IgG, IgA, IgE, etc. And so given that our CD8 T cells in autoimmune disease kind of look like CD4 T follicular helpers, and they do seem to encourage B cell antibody responses, we wanted to know whether or not the CD8 T cells were present here in this germinal center. And in order to do this, we did some imaging. So did a lot of confocal. And so what we found was that in immunized wild type mice, so these mice were immunized with KLH plus incomplete for its adjuvant. What we found is that CD8 T cells are not present either in the germinal center or the surrounding B cell follicle in these mice. But in our autoimmune model, these cells are present in the surrounding follicle, which is going to be defined as IgD positive. And then in the germinal center, which is a PNA stained region that generally does not have IgD, which is going to mark our naive B cells. And so we see CD8 T cells both in that germinal center area and in the surrounding B cell follicle. And this is significant when looked at all of the ones that I did.
Hi, Genevieve.
Yes.
Okay, Genevieve, thanks a lot. And now we can continue.
All right, awesome. And so yeah, from this data, we at least managed to identify that there are CD8 T cells that can, in autoimmune disease, localize to the B cell follicle and the germinal center. From there, we kind of wanted to further, one, in this particular staining, we don't know whether or not these CD8 T cells express CXCR5. We assume they probably do, because how else did they get there? But we wanted to confirm that, as well as kind of try to identify, were they localized to a particular region of the germinal center, i.e. light zone versus dark zone. And so in order to do that, we did more staining. And so we keep CD8, PNA, and IgD in order to look for the follicle and then the germinal center region. And then also looked at CXCR5 to mark those CXCR5 CD8 T cells. Again, CXCR5 should allow them to get into the B cell follicle and the germinal center. But also CXCR4, which is a chemokine receptor that on B cells generally marks dark zone localization, So we were just kind of curious as to whether or not these CD8 T follicular cells might be expressing this marker that would allow them to be in the dark zone primarily of the germinal center. And so what we found was that in the germinal center CD8 T cells, most of these CD8 T cells were expressing both CXCR5 and CXCR4. And the combination of these two does suggest that those CD8 T cells may be preferentially locating into the dark zone of the germinal center, a region that generally CD4 T follicular helpers aren't found in. So that was really interesting to us, and something we're hopefully my old lab will continue to follow up on to kind of try to figure out why might they be there. And then while this was also true in the follicle, that most of this, or the highest expression level of these CD8 T cells was on CXCR5, CXCR4 double positive cells. This was a lot more varied in expression in the follicle. And I suspect that this is due to cells kind of entering and leaving regions within the follicle and germinal center. So there's kind of more fluctuation of chemokine receptor expression inside the greater follicle area than in the germinal center proper. And so overall, this is really interesting. And so it did show to us that there are CD8 T cells, particularly in autoimmune disease, that can localize to the B cell follicle and the germinal center, and that they might preferentially be localizing to the dark zone, which was really interesting. And so from there, we wanted to kind of start to delve into kind of what do these cells look like? What possible functions are they doing? And how might they be mediating those functions? So we started with looking at their transcription. So we isolated CD4 T follicular helpers and CD8 T follicular cells from IL-2 knockout mice day 15 to 16. So generally these mice in our lab succumb to disease by day 21, and most of them are fairly sick by day 18. So we were aiming to get cells kind of before the worst of kind of disease progression, so we could try to capture some of the early changes between these two cells, while also still choosing a time point in which, from some kinetics data, there was still a decent number of CD8 T follicular cells present. And then we did some Illumina RNA sequencing, and then differential gene expression analysis to kind of try to decide what kinds of things were different between these two cell subsets, primarily because we wanted to know whether or not these CD8 T follicular cells expressed anything unique from their CD8 T follicular helper counterparts that, at least so far, they looked very similar to. So we looked at a variety of genes, obviously, since that's what you get to do with RNA sequencing. So we looked at markers of follicular cells, kind of more general effector molecules, particularly for CD8 T cells like granzymes, perforin, interferon gamma, exhaustion markers, particularly since this is a chronic antigen situation. These cells could be expressing exhaustion markers, and then memory markers. And so not surprisingly, CD8 T follicular cells do express a decent amount of follicular markers, things like BCL6, ICOS, CXCR5, things that we had confirmed or seen already from flow cytometry data at protein level. What we can see based on the RNA expression levels is that generally these CD8 T follicular cells, while they do express a lot of these markers, express them at lower levels than their CD4 counterparts. So while they do express follicular markers, they're kind of less follicular than their CD4 T cell counterparts. And as I noted from our previous data that we looked at by protein level that I didn't really show, these cells also express markers that kind of mark them as a more traditional CD8 T cell, like granzymes and perforin. They also express a decent number of exhaustion markers, like LAG3 and TOX and TIM3. But they also express memory-like markers, which is, while at the time might have been a little bit surprising, with a lot of the other work from other labs looking at CXCR5 positive CD8 T cells, this might be less surprising now, since some labs suggest that CXCR5 is a marker for memory precursor cells. So it's possible even in our autoimmune model that they might be kind of a little memory-like. So overall, this did suggest that these CD8 T follicular cells have kind of a helper-like and memory-like phenotype, while retaining more cytolytic markers and phenotype.
We have a question
Yes.
from one of our viewers. The question is, what could be the TSC doing in the dark zone?
So what we've kind of thrown around a lot of ideas, and we haven't really tested them yet, since when I got the data showing that they were in the dark zone, I was pretty much out of my program already. But we threw around ideas that possibly the reason that these CD8 T follicular cells might localize to the dark zone is that they might be potentially giving signals to B cells in the dark zone, either to like help additionally with proliferation or the somatic hypermutation. But given that, at least for the field with like CD4 T follicular helpers, there's really not a role for them in the dark zone because the CD4 T follicular helpers sit on in the light zone. It's hard to say for sure what it could be. One other thing we were thinking it could be is that because these CD8 T follicular cells retain markers of cytolytic cells, and while we have not had a whole lot of success testing whether or not these cells are actually capable of killing target cells in vitro, other studies of CXCR5 CD8 T cells suggest that they could actually kill target cells. One idea is that the dark zone of the germinal center, while a lot of B cells do proliferate there, there is some apoptosis that occurs there, as like if B cells get to a point where they mutate their antibody to such an extent that now it can't form a functional protein, those cells will die. And so one thought is that maybe the CD8 T follicular cells in the dark zone might help facilitate killing off like bad B cells. But again, there's definitely further testing that we would need to do that we haven't gotten around to doing yet.
Would you think they might be giving a shortcut route for these low affinity B cells to get out because they would be just giving survival signals, so they don't need to go to the light zone anymore?
Yeah, that might also be possible that maybe they might encourage cycling without going back to the light zone, just like cycle in the dark zone. One other thought is potentially that they might help with getting cells leaving the dark zone directly rather than like going back to the light zone and leaving as like memory or plasma cells. I don't think I have it in here, but in our JI paper, we have some evidence that suggests that CD8 T cells from our autoimmune mice can influence or increase plasma cell generation. And so that could also be one, is that maybe they're just like shooting off B cells from the dark zone into plasma cells without them having to return to the light zone and get those signals from CD4 T follicular helpers. So there's a lot of possibilities and a lot of things that need to get kind of hammered out with it.
It's such an interesting point here. You got a new cell type and could be doing so many new things.
Yeah, it was probably the most exciting piece of data I got, and I was so glad I got it. I mean, it's kind of sad I got it kind of towards the end of my PhD work, because then I was like, well, I'm leaving, so that's on you guys. But it was nice to end with okay, that's kind of cool. All the work I just did was worth something. Particularly all the imaging, which involves sitting in a dark basement for hours on end. So, yeah, with no phone cell service. So, fun times. Okay, so Okay, so yeah, now I've shown these cells localized to the B cell follicle, and that they seem, at least transcriptionally, to have kind of both helper-like and memory phenotypes, while kind of retaining markers of cytolytic activity. So from there, we wanted to look at their cytokine protection, primarily cytokines IL-4 and IL-21, as these are key cytokines produced by CD4 T follicular helpers, and then kind of further whether or not these CD8 T follicular cells are contributing to B cell function. So here we are back in the Germinal Center. So now we have CD4 T cells, we have B cells here in the Germinal Center. Now we also know that there are these CD8 T follicular cells that can interact with, at least to the extent of being able to get into the Germinal Center and the surrounding B cell follicle, and particularly might be able to localize here to the dark zone. And some of our data from our JI paper, which I'm not showing today, suggests that they might not contribute to the frequency of Germinal Center B cells. So they might not help with cycling, although again, that might just be with light zone cycling, it's hard to say, but can contribute to plasma cell generation. One thing we wanted to then assess is, since CD4 T follicular helpers can produce cytokines like IL-4 and IL-21, we wanted to know whether or not these CD8 T follicular cells could also produce these cytokines, particularly whether or not they're producing them in like the Germinal Center or the surrounding B cell follicle. For these, unfortunately, we could not assess dark zone, just limitations of the imaging, but I'm hoping in the future to be able to try to do that anyway. So just from our flow cytometry data, we kind of first looked broadly at some cytokines. And so first off, our CD8 T follicular cells in comparison to here, the non-T follicular cells mean CD8 T cells that are negative for both PD1 and CXCR5. So there are a little double negative population of CD8 T cells. So first off, in general, the CD8 T follicular cells in our autoimmune model produce more cytokine than their non-follicular counterparts. And those cytokines that they are making do seem to be largely things like IL-21 and IL-4 here in the white, and then the combination of those two here in the blue. But they also produce like IL-10 to varying amounts with these other two cytokines. And to a much bigger extent than their non-CD8 T follicular cell counterparts. So that's cool. They make these cytokines, but again, we wanted to know whether or not they were making these in regions that are going to be helpful or promoting B cell functionality. So went back and did some more imaging. And so again, our Germinal Center is IgD kind of negative with PNA staining. So the IgD is our surrounding B cell follicle. And so we looked at IL-4 and IL-21. And so in general, there's lots and lots of IL-4 and not as much IL-21. And what we found was that in general, those CD8 T cells, both in the Germinal Center and in the follicle that expressed at least one of the two cytokines I was staining for here, were largely producing IL-4 alone, and to potentially a varying, a lesser degree, but still a decent degree, IL-4 in combination with IL-21, but not very many of them were making IL-21 alone. There were, as you can see, a decent number of CD8 T cells from this imaging that weren't expressing either IL-4 or IL-21. And that might be that these cells were producing other cytokines that I was not at the time staining for, like interferon gamma, which from our model, well, both C4 and CD8 T cells make in abundance. So, but this was nice, and it helped suggest that these functional CD4 T follicular helper cytokines, IL-4 and IL-21, were being produced by CD8 T cells in regions where they could act on B cells. So we wanted to then start to assess whether or not these cytokines, particularly being produced by these CD8 T cells, could or were influencing B cell function. And so to do this, we did a in vitro indirect co-culture assay. So we sorted out IL-2 knockout CD4 T follicular helpers or CD8 T follicular cells, plated them with T cell stimuli for 3 days, and then collected their supernatant. So any of the things that they were secreting, all those cytokines, were then plated on top of naive wild type or IL-21 receptor knockout B cells. And then they were provided with B cell simulation, and then these cells were assessed for expression of AID, which I will mention in a moment as to what that is, and antibody levels and class switching. So what we found for the antibody level, at least for this assay, when we were looking at the contribution of IL-21, we found no difference. That whether or not the B cells could see the IL-21 made no impact on the antibody class switching, because here we're looking at IgG. Even for the C4 T cells, where we expected there to have been a difference. And so from this, really, we assumed that the in vitro co-culture, at least for the antibody levels, that the difference in antibody levels was probably too minor to pick up by ELISA the way we were doing it, which is fine, because we also wanted to look at the B cells more directly. And so in the Germinal Center, when CD4 and T cells interact with B cells, among the things that happen to B cells from this interaction is that they will induce expression of AID, or activation-induced cytidine deaminase. And this is a protein that is responsible for carrying out somatic hypermutation and antibody class switch recombination. So it replaces, or it causes Cs in the DNA to become uracils, and then those uracils trigger DNA repair mechanisms and they get replaced. And so this protein is really important for what happens to B cells during this reaction in terms of like their affinity maturation and their ability to class switch. So we wanted to assess whether or not CD8 T follicular cell cytokine, at the very least, could influence the production or expression of this protein. And so when we looked at our IL-21 receptor knockout B cells, there was a decrease in AID expression in B cells when shown or given CD4 T follicular helper supernatant, a difference that was not seen when provided CD8 T follicular cell supernatant, which suggested that the AID levels in the B cells, at least from CD8 T follicular cells, was not being influenced by IL-21. And part of that might be that the CD8 T follicular cells just aren't producing as much IL-21 as their C4 T follicular cells, or T follicular helpers. And so that might be why. But we also looked at IL-4, that cytokine that, at least from the imaging data, CD8s were making primarily within the Germinal Center. And so in this case, we depleted or neutralized out IL-4. And so we get a decrease in AID both from C4 T follicular helper and CD8 T follicular cell supernatants. And this suggested that IL-4, at least from CD8 T follicular cells, was impacting B cell AID expression, and that in turn could influence antibody class switching or somatic hypermutation in these B cells.
Genevieve, this AID expression, are you looking at it inside the T cells or the B cells?
This is looking at it in the B cells. So after,
Okay.
After the B cells get plated with supernatant, 3 days later, I pull the B cells off of the plate and then assess AID expression.
Okay.
in at least what B cells remain there, because these are mouse B cells and they don't necessarily like being in culture for that long.
Okay. So the x-axis tells you what they were co-cultured with. Okay.
Yes.
Okay.
And yeah, I'm not showing it, but we also did like just wild type CD4 T cell supernatant as well. And that doesn't generally do a whole lot because they're not really making anything, but okay. So it kind of, in summary, from all of this data, what we know about the germinal center response before all our work was that there are CD4 T follicular helpers that come in and can provide B cell help to allow them to cycle through the light and the dark zone and differentiate into memory and plasma cells. And what we have found with our work is that there are these population of CD8 T cells that express CXCR5 that can also get into the germinal center and the surrounding B cell follicle, including potentially the dark zone. And that these cells, some of our data suggests might not help with germinal center B cell cycling, but instead help with plasma cell differentiation. But that these CD8 T follicular cells are producing cytokine, both here in the germinal center and in the surrounding B cell follicle, primarily IL-4, but to at least a little bit of IL-21. And that at least some of this cytokine secretion influences AID expression in the B cells here. And whether or not that cytokine is doing anything to dark zone B cells, we still need to kind of figure out like what would those even do in B cells in the dark zone. And then potentially, since there is a possibility of CD8 T cell interactions with B cells outside of the germinal center proper, that these might also help encourage plasma cell differentiation without the rest of the germinal center. And so with that, I will acknowledge my former lab, since that's where all this work was being done. So Katrina, who, was responsible for me being in the lab to do all of this work. Kristen Valentine was Dr. Kristen Valentine was the other graduate student in the lab who I collaborated with for all of this work, and is co-first author on the paper I showed stuff from. Oscar Davalos was the graduate student who did all of the sequencing analysis. Our great collaborators at UC Merced that helped us. So David Garano is like our flow core guy. So he helped with any questions we had for all of our sorts and stuff. Emily Slocum in the animal facility. Anand Subramaniam, who is in the imaging core. So thank him. I'm very grateful that he let me use his confocal microscope. And then of course, our wonderful funding sources to allow us to do all of this work, because all this stuff costs money. And then thank you all for listening to me talk about the paper that I haven't thought about in a little while.
Thanks a lot. Yeah, thanks a lot for joining us today. For me, it was introduction to a completely new concept, because it's more almost paradigm breaking. You would think that there's only CD4 is responsible for antibody helping, but no, not anymore.
Yeah, it is really interesting, and it's one of the things that I think has been a struggle with it. Whenever we go to kind of meetings with germinal center and like C4 T follicular helper people, is their thought is like, well, so what? Like, so what? CD8 T cells can do this, because at least a lot of what we've shown so far is that they basically just look and function like CD4 T follicular helpers. And while it's cool that CD8s can do that, what's the point if all they're doing is like the same thing that CD4 T cells already can do? And so that, I think, is where our dark zone localization stuff will start, especially once we start to figure out what they're doing in the dark zone. We'll start to kind of help tease apart what are the CD8s doing that CD4s can't do. Like, what are they uniquely contributing to the germinal center response?
Do you think there's any they're induced by an antigen?
So we do know that the CD8 T cells are definitely well, so we've had some trouble with doing like cytotoxicity assays, but we are pretty sure that they are antigen specific. Some of our in vivo studies we've done, we looked at like antigen specific antibody production just to double check, and it like, okay, cool, there is at least some antigen specificity. It's hard to know for sure because in our model, other than knowing that red blood cells are being targeted, we don't really know what the antigen is. Our model is just not as clean as models where you know those, but it's fine. But one of the things that we've wanted to do for a while and try to figure out was whether or not, like, are they for sure responding through like MHC1, or like, is that how they're because C4 T follicular helpers in the germinal center see antigen on B cells through MHC2, and so the thought would be then that the CD8s would be seeing antigen that the B cells are presenting on MHC1, and so we're I think we've played around with ideas on like how exactly we would determine for sure. I just don't think we've gotten around to some of those experiments.
Yeah, like you said, in the wild type mice, they were not there, almost absent. So I wonder if they're immunization dependent or just completely in the context of autoimmune diseases. That would be a fun thing to know about in future.
Yeah. Some data from from our Journal of Autoimmunity paper that I didn't show because I think it was really mostly like we were trying to preempt reviewers being upset at our model. We did do some kind of like double knockout systems to try to kind of tease apart what is required for CD8 T follicular cells to develop. And they do require CD28. They don't necessarily require interferon gamma. They don't necessarily need IL-21 to be, or IL-2 to be absent, because we can also see them in the scurfy deficient mice. Although what we did find is that if you deplete or get rid of IL-2 in these IL-2 sufficient autoimmune models, they do increase. And part of that is that without IL-2, there's just massive lymphoproliferation. And so it's like, okay, yeah, the IL-2 might be inhibiting their production, but it's also more than likely it's like the lack of IL-2 is just allowing all T cells to expand. And so then you get more of the CD8 T follicular cells because there's just more T cells in general. And so they definitely seem to they require co-stimulation because they require CD28. They don't necessarily require interferon gamma, but they only arise in like systemic autoimmune diseases and not tissue specific ones. So it's kind of, it's really weird. And it might just be that like they just need chronic inflammation. And if we could manage to achieve that, maybe like in our immunization settings, maybe we would see them. Or like some of the work from like exhaustion in like chronic viral diseases and stuff are showing that, some of the requirement for like exhausted cells and stuff is like that it's chronic antigen, not chronic inflammation. So it's hard to say which of those two in our model is more important because we can't get rid of the antigen in autoimmune disease.
Yeah, I think your work has opened doors for at least the next 4 graduate students in your previous lab.
Yeah, it should be fun. Let's see. There she is. Christy is I didn't add her to my but yeah, Christy here is the graduate student, I think, mostly pulling kind of some of the work off of to continue on. Her interest is maybe less B cell stuff than mine was, but hopefully she'll also find some interesting stuff about these CD8 T cells. So.
We have one more question from our viewers. So if the Germinal Center was devoid of CD4 T cells, do you think that CD8 T cells could rescue the functions of CD4s?
That is a really good question, and it's a really interesting one. So, and it's not one that we haven't thought about. So, in we've done some of our in vivo work. We took TCR alpha knockout mice and then transferred in our autoimmune CD4 CD8 T cells. For those studies, we had to do bulk because for some reason the follicular populations like just weren't enough when they were sorted and transferred in. Like they just didn't survive well without like other T cells around. But what we found was that you really did not get germinal center B cells or antibody production if CD8s were transferred alone. They needed CD4 T cells to also be transferred. And so that made us think that there's the possibility that the CD4 T cells need to at least start the germinal center, like they need to get the germinal center started, and then the CD8s can join in once there's like a mature germinal center, since the germinal center does progress in stages. But it's hard to know that for sure. And one thing that would be interesting to do would be to like deplete out the CD4 T cells once the germinal centers are like formed and functional, and see whether or not the CD8s can maintain a germinal center once the CD4 T cells are gone. Like, do they only require the CD4 T cells to be there when the germinal center forms, or do they need CD4 T cells to be there the whole time? That's something that would be really interesting to look at. And it is weird because some data from like the early 2000s in rheumatoid arthritis did show CD8 T cells in ectopic germinal centers, and that the CD8 T cells were required for ectopic germinal centers. And so maybe there could also be a difference between like ectopic versus like lymphoid organ germinal centers in terms of the requirements for CD4, CD8 T cells for the formation and maintenance of the germinal centers. And that's something that would also be interesting to dissect.
Right, thanks a lot for that. And I think this would, unless if you guys have any questions before we wrap up the session. Okay, thanks a lot, Genevieve, for coming here. It was a great meeting and a good learning experience for us all. For our audience, we'll see you again in 2 weeks with our next Journal Club. Until then, goodbye.